Hello students, we will be starting our next chapter of biology, that is reproduction in plants and animals. Now, the first question arises, what is reproduction? We know that the reproduction is one of the most important characteristics of life. Why? Because it maintains the continuity of our life on earth. We are because of this process of reproduction. So what is reproduction? It is the process by which living organisms produce new individuals of their own kind. Modes of reproduction. Now, there are two main modes of reproduction. One is the asexual reproduction and another is a sexual reproduction. Now, in case of asexual reproduction, only a single parent is involved and there is no fusion of male and female gamete cells. There is no fusion of the male and female reproductive cells called as gametes. So, reproductive cells are called as gametes. So, asexual reproduction is a type of reproduction which does not involve the fusion of gametes, and the offsprings are exactly the same of the parents. So, in case of asexual reproduction, only a single parent is involved and there is no fusion of male and female gametes. So, as only one parent is involved, so the offsprings are exactly same as the parent. Sexual reproduction. In case of sexual reproduction, the two parents, the male and the female, are involved. And the male produces the male gamete, that male gamete is called as sperms, and the female gametes are called as ova or egg. So, sexual reproduction is that reproduction in which two parents are involved and the offsprings are produced by the fusion of male and female gametes. Male gametes are called as sperms and the female gametes produces the female gamete, and the female produces the female gametes, and the female gametes are called as eggs or ova. Next, asexual reproduction in plants. Asexual reproduction in plants takes place by few processes, and they are fragmentation, spore formation and vegetative propagation. Fragmentation, spore formation and vegetative propagation. Now, what do you mean by fragmentation? So, it is a method in which an organism breaks up into two or more parts, and the two or more parts are called as fragments. You can see here the parent cell has divided into two fragments, and these fragments then grow into new individual. This type of fragmentation process is very common in plants like Spirogyra. Under favorable condition, algae also grow and multiply by fragmentation. So, what is fragmentation? It is a method in which an organism breaks up into two or more parts called fragments, and these fragments then grow into new individual. Spore formation. Some lower organisms like bread molds, ferns, they undergo this type of spore formation. They develop rounded or oval structure on their body, and that rounded or oval structure on their body is called as spore sacs, which contains spores, and they are released when the spore sacs opens and they develop into a new plant. So, we can see spore formation process in case of bread mold or ferns. Next, vegetative propagation. It is the development of new plant from the vegetative parts. Vegetative parts means root, stem or leaf of a plant. So, it refers to the process of plant reproduction whereby a fragment of a parent plant is taken in order to produce more plants. It's common in plants which do not produce seeds or whose seeds cannot germinate. So it is also a form of asexual reproduction. Why? Because there is no involvement of reproductive structures. Yes, so sorry. So asexual reproduction in plants takes place by fragmentation, spore formation and vegetative propagation. Fragmentation means it's a method in which an organism breaks up into two or more parts, and the two or more parts are called as fragments. Spore formation, in this parent plant generates thousands of spores which individually grow into a plant, and these spores are microscopic. You can see here in this figure the bread mold is shown, and this figure is showing the spores there. The sporangiophore is there. It contains the sporangium, and the sporangium contains the spores. Vegetative propagation, it refers to the process of the plant reproduction whereby a fragment of a parent plant is taken in order to produce more plants. And it is common in plants which do not produce seeds or whose seeds cannot germinate. It is also a form of asexual reproduction because there is no involvement of reproductive structures. Now, there are two methods by which vegetative propagation takes place. One is the natural method and another is the artificial method. In case of natural methods, in some plants, reproduction by vegetative propagation takes place naturally from different parts. And what are the different parts? Different parts includes stems, leaves, roots of the plant. So first, from underground stems. First reproduction is from underground stems, and this type of reproduction occurs through bulbs formation, rhizomes, tubers and combs. So tuber in potato. So potato, they reproduce by tuber formation. The first picture of the potato, that what is shown, you can see some marks are there. They are called as the eyes or scars, and the vegetative buds present in these eyes, and reproduction takes place from that buds. On germination, the eyes give rise to new plant. Next, rhizome, that is found in case of ginger. So in ginger, the short branched underground stem, that is called as rhizome, and that is used for reproduction. Banana also grows from rhizomes. Next reproduction is corm, which is found in case of gladiolus. Gladiolus is a flowering plant. So the corm or thickened round underground stem of gladiolus is used for vegetative propagation. Bulb, that is common in garlic or lily or tulip. Bulbs of onion and in case of garlic also, they are used for vegetative propagation. Next is from aerial stems. So in plants like strawberry or oxalic, a long horizontal stem, it grows over the soil surface. And that long horizontal stem that grows over the soil surface, that is called as runner. And wherever the stem bends and touches the soil, it develops roots and buds at the point of contact with the soil. You can see here, this is a strawberry plant. Some stems are growing. So that stems, wherever they touch the soil, roots grow over there and bud develop into new plant. It is found in case of lawn grasses also, in case of mint also. New shoots and roots arise from the nodes producing new plants. Next process is from leaves. That is common in case of bryophyllum. So, in this case, buds are present in the notches on the margin of leaf. The bud grows on the margin of the leaves, and plantlets develop from that buds. When they get detached from the parent plant, a new plant develops. Next, from roots. This is common in case of sweet potato or dahlia. These plants have swollen roots and these roots store food. These buds give rise to new plant on getting discharged. So these are the few processes of vegetative propagation. Next, tissue culture, that comes under artificial method. So previous one was the natural method, that is vegetative propagation takes place by two processes, that is one is natural method and another is the artificial method. Natural method one is the from underground stem, from aerial stems, from leaves and from roots. These are all the natural methods. And artificial method is a tissue culture method. So some vegetative propagation is a fast method of reproduction in plants and it has been adopted by human beings. And it is carried out by human beings to grow plant in an artificial medium. And such propagation is done by tissue culture. So what is this? It is the growth of the tissue or cells in an artificial medium in a test tube. And thousands of new plants are produced from a small piece of tissue by this process of tissue culture. What happens? First, the shoot tip is cut from the main plant. At first, the shoot tip is cut from the main plant. Then, it is divided into many small pieces of tissues. Each piece is then put in a nutrient medium, in a test tube, and a germ-free environment is maintained. Then, the tissue grows into an unorganized mass of cells, and that unorganized mass of cell is called as callus. At first, small pieces of plant tissues are taken from one plant. It has been kept in a nutrient medium and it has been seen that the medium should be a germ-free one. After that, the tissues grows into an unorganized mass of cell and that unorganized mass of cells are called as callus. Small part of the callus are transferred to another nutrient medium. So there is a new growth medium. Small parts of the callus gets transferred to new growth medium. And they induce the formation of root and shoot. And thus new plantlets are formed. And these plantlets are then transferred to the soil and ultimately they develops into a new plant. So all of you understood this tissue culture process? I am repeating once more. At first, the shoot tip is cut from the main plant. It is then divided into small pieces of tissues. Then each piece is put in a nutrient medium, in a test tube. That is also a germ-free environment. The tissue grows into an unorganized mass of cell. That is called as callus. Small part of the callus is transferred to next medium. That is another growth medium that contains growth hormones. And that result in the formation of root and shoot. And finally develops into plantlets. And the plantlets are then transferred to the soil and ultimately that develops into new plant. Thousands of the new plants are produced by this process. Like this, some orchids are there, then chrysanthemum, they all are produced by this method of tissue culture. It is an artificial method. Now, some home assignments for you. Question number 1. Give 2 examples of each of the following. Plants reproduced from underground stem. Plants reproduced from aerial stem. Plants reproduced from leaves. Plants grown by tissue culture. So students, I hope all of you understood till here. The next portion we will be discussing in our next video. Thank you.